Okay, so this is a plant, and now we're zooming into the leaf to where the chloroplast is. The beginning of photosynthesis starts with the light reactions, which takes place in the thylakoid, while the Calvin cycle takes place in the stroma, but we'll get to that later. Okay, so to start, a photon of light strikes a pigment molecule in a light harvesting complex, boosting one of its electrons to a higher energy level. Then this electron is eventually transferred from the excited p680 to the primary electron acceptor, while an enzyme catalyzes the splitting of a water molecule into 2 electrons, 2 hydrogen ions, and an oxygen atom. And with these hydrogen ions, they diffuse from the thylakoid space back to the stroma through ATP synthase complexes, with proteins flowing down their gradient causing the rotation of ATP synthase, making ATP from ADP an inorganic phosphate. And each photoexcited electron passes from the primary electron acceptor of PS2, or photosystem 2, to PS1 via an electron transport chain. As electrons pass through the cytochrome complex, the pumping of protons builds a proton gradient that is subsequently used in chemiosmosis. Meanwhile, light energy was transferred via light harvesting complex pigments to the PS1 reaction center complex. Photoexcited electrons to pass through in a series of redox reactions from the primary electron acceptor of PS1 down a second electron transport chain through the protein ferredoxin. However, this chain does not create a proton gradient and thus does not produce ATP. Finally, the last stage of the light reactions, the enzyme NADP+ reductase catalyzes the transfer of electrons from FD to NADP+. However, it will require 2 electrons for its reduction to NADPH. Now that we've produced ATP and NADPH, we can now move on to the Calvin cycle. Okay, now welcome to the Calvin cycle. And from the light reactions, we brought ATP and NADPH to the Calvin cycle with us. The first phase of the Calvin cycle is carbon fixation and is catalyzed by the most abundant protein in chloroplast called rubisco. Phase 1 of carbon fixation then ends with the product of 2 molecules of 3-phosphoglycerate for each CO2 fixed. That's when we move on to phase 2, reduction. That's when a pair of electrons donated from NADPH reduces 1,3-biphosphoglycerate, which also loses a phosphate group, becoming G3P. And G3P is a sugar, the same 3-carbon sugar found in glycolysis by the splitting of glucose. Also to note that for every 3 molecules of CO2 that enter the cycle, there are 6 molecules of G3P formed. One turn of the Calvin cycle produces 6 molecules of G3P. However, 1 molecule exits the cell to be used by the plant cell, but the other 5 molecules must be recycled to regenerate the 3 molecules of RUBP, also known as ribulose bisphosphate. Finally, the last step of the Calvin cycle is phase 3, regeneration of the CO2 acceptor, ribulose bisphosphate. The cycle uses up 3 more molecules of ATP to rearrange the carbon skeletons of 5 molecules of G3P into 3 molecules of ribulose bisphosphate. Once formed, the cycle is ready to receive CO2 again, and the cycle continues. For a quick overview, for the net synthesis of 1 G3P molecule, the Calvin cycle consumes a total of 9 molecules of ATP and 6 molecules of NADPH. The light reactions regenerate the ATP and NADPH.